Thanks for hosting this. So I'm Hazrat. I'm a lecturer at the University of Stirling, and here I teach computer science, basically deep learning, AI. So I work in AI for medical imaging, and we are going to talk about some of the things that we have been doing over the last few years in the space of generative models in medical imaging applications. So let's start with the slides. So this one is actually better, this title slide, so I put it here. This one is from SICSA. Okay, so today we are going to talk about some of the work that we have been doing related to motor unit segmentation in human body muscles. We are also going to talk about a disease known as atherosclerosis, which is basically related to the accumulation of plaque in the arteries. And then we are going to also talk about some of the applications in image to image translation things. So we will specifically talk about CT to MRI, and we will also touch upon applications in super resolution. And although it's not listed here, but hopefully if we got time, we will briefly touch upon some work related to diffusion models. Before we start, a bit of a disclaimer here. So here, although the title says generative AI for medical computer vision, but here generative AI does not just mean LLMs. Basically, we are talking about generative models such as generative adversarial networks and neural diffusion models. So you won't see anything related to LLMs here today. Okay, so the first topic that we are going to talk about is the segmentation and studying of motor units in human muscles. And this was a project that I undertook at Umea University in Sweden. By the way, Umea University got into the spotlight at that time. So back in 2020, one of the chemistry Nobel Prize winner was actually someone who worked at university before, Emmanuel Shapinshire. So she actually worked at UMU before moving to her next institute. So this project that I'm going to discuss was led by Christer. So it's a giant project related to ultrasound imaging methodologies. So we are going to talk about a specific case study here. So when we study human muscles, typically the way they would do it in clinics is basically through invasive approach by doing actually inserting needles under the skin. Yeah, is there some? I hope I'm audible. So there.
You're fine. I think it's just a bit of background noise.
Okay, okay. So typically the way they study it is through inserting, these needle electrodes under the skin to record the electrical activity. There's also another approach to do it is through, surface electrodes by connecting the electrodes to the skin. Uh, One of the common limitations in these methods is that although they are quite effective, but they provide you very limited field of view. So if you look at the needle method, basically you are looking at a very, small point at a time. And likewise for the surface electrode, again, you are covering a very small part of the skin. So we believe that ultrasound can record a much broader view and it can be a good alternative. And another benefit that it brings is that it is non-invasive. You don't, rely on actually inserting a needle under the skin. So in terms of this muscle activity, if we represent it with the help of some visuals, actually the muscle activity is something that the muscle will basically expand and contract. So this 3D kind of visualization shows that phenomena that we have this muscle expansion and contraction. Okay. This is another way of representing it. So we will have this muscle velocity. It will increase and decrease, increase and decrease. And this signal is something that we call is the twitch signal. Okay. So what we see here is the expansion of the muscles, the contraction of the muscles. In ultrasound, it will be simply a 2D representation of that. So we can now relate it that the muscle is basically expanding. So in ultrasound, we can see that as the time passes on, in 2D, it will basically have an expansion in this area and contraction in the area. One of the common challenge here is that some of these motor units are actually overlapping. Okay, so this is quite a challenge. So what we do here is we develop a deep learning pipeline. We thought, okay, let's try AI. So we developed a deep learning pipeline and we used a CNN, basically a 3D CNN, along with a mass RCNN model to basically perform the segmentation of the motor units. And then we also use CNN followed by an RNN to track the time domain signal as well. So as I said, that the challenges here is that these motor units are overlapping and they have changes in the intensity because of the expansion and contraction phenomena. Okay, so this is something that we just discussed. So there was some previous work in the group that used signal processing techniques such as blind source separation, uh, but these techniques again were able only to basically track a single motor unit and they couldn't move beyond a single motor unit. So when you increase the number of active motor units, these techniques actually struggled. So our hypothesis is that we can use this we can use a deep learning pipeline to perform the identification of these individual motor units, okay, by some sort of extraction of information from the expansion and contraction, and then use this to track the time domain signal as well. So here is the pipeline. I won't go into much details because these figures can be quite boring for some of us. So this is just a 3D neural network. Okay, it extracts information, it extracts features, and then we use them along with mass RCNN to perform the segmentation. Okay, and here we are talking in terms of the data, we have around 10,000 image sequences, okay? And we use 40,000 out of these, okay? And this training process overall was done on a single GPU, so it took around 60 plus hours. Followed by the segmentation of the motor units, we also do the tracking of the time domain signal, just represented here with the help of this visual. So here is the network implemented for the time domain signal tracking. So it uses GRU. Okay, again, the 3D signal, the 3D CNN is simply used to convert the data into like one dimensional representation, which is then fit to the GRU to basically perform the training. Okay, so I'll skip this and move to the results because these are interesting. So here what we are looking at is something that we have here an activation of 5 motor units. So we are looking at 5 motor units on the left side. Okay, and we can see that the model is able to detect these motor units. On the right side, we also look at the time domain signal of this activation that we call as the twitch trend signal. So we can see that the model is doing fairly good. It can trace the time domain signals quite nicely. Even if it is not like tracking exact shape, it is still able to track the rise and fall in the signal. But here is the challenge. So when we increase the number of motor units beyond 5, actually here we are using around 16 motor units. Okay, so now again we are doing, that model is doing quite good, but the challenge that for the motor unit number 6, I think is easy to spot in this figure. For the motor unit number 6 and number 8, we are not able to basically perform the detection. And also because we couldn't detect the motor units, it couldn't detect the time domain signal as well. Okay. And the obvious reason for this is the kind of, overlapping of these motor units. So the partial overlap might be fine, but a complete overlap is basically affecting the performance of the model, as we can see in case of motor unit 6. And just in case you are interested in how these motor units look like in actual ultrasound representation, this is what we are looking at when we have five active motor units. So it's not that, like it's not that high contrast data that we are looking at. And if you increase it to 15 MUs, 15 motor units, it becomes even more complex. So yeah, this was something that we did. We still feel that like it needs some improvements. Of course, it wasn't able to do very good on the overlapping motor units. There is some proof that it can help, but we need to look beyond these methods to basically be able to detect as many active motor units as possible. Okay, so I'm moving to the next project here is related to atherosclerosis disease transformation. So atherosclerosis is a disease where the plaque would accumulate. I'm sorry about this. So it's a disease where a plaque would accumulate on the inside of the arteries, okay? And this plaque can eventually lead to stroke or heart attacks. So the way, and this plaque is basically on the carotid arteries near our neck, okay, in the neck region. So the way clinicians would study this plaque is by taking ultrasound images of the carotid arteries. And the way they do it is they actually record ultrasound images from 4 different angles. So for each single case, you have like, 4 ultrasound images coming from recorded from 4 different directions. And the more the accumulation of the plaque is, the high risk the individual is. And then there is an interest to study this accumulation of plaque in certain individuals, along with other different clinical factors, socio factors like living style, lifestyle, eating habits, smoking, non-smoking behavior, and so on. What we are looking at now on the left side is an ultrasound image of someone who is at this stage a healthy individual, okay, with almost no disease, no atherosclerosis. So if we look at this region that I'm trying to highlight with the help of this pointer. So if you look at this region, there is no plaque, very healthy individual. Same for the second image. On the right side, we are looking at an ultrasound image of someone who is not healthy, who has kind of pronounced atherosclerosis. There is a lot of plaque already accumulated in the carotid artery. So this was a data that was recorded in Sweden, and they would study it for the passage of 3 years and then for another 3 years. So the same individuals would be called upon, to appear for a follow-up checking and kind of recording the ultrasound data again and then comparing with the original ultrasound images. So it was like a large project that they were doing. So the project was called Visa Project. They had registered 3,500 healthy individuals, and basically these individuals were to come for a follow-up visit, after 3 years, so that their ultrasound can be recorded again. What we did was we picked some of the data from this huge data set. So we picked a few selected images, around 2,300. That we believed were healthy individuals with almost no atherosclerosis, and the criteria is basically the thickness of the intima media, basically the accumulation of the plaque that is visible in the ultrasound images. So we picked those images or those individuals where we only had, a thickness of 0.3 to 0.5 millimeter, and clinically they are referred as the healthy individuals. And then we picked few images, around 2000, from those cases where we had pronounced cases of atherosclerosis. So here the criteria is that the thickness is greater than 1.5 millimeter. So we collected this data, we picked up this data. did the regular 80 to 20 split, and the goal is that we want to use an AI model to go from the no disease scenario to the pronounced disease scenario. So the way we did it, we trained a generative adversarial network again. We know that gains are good at domain to domain translation, so we treat this problem as a domain to domain translation. So you might have seen the application of gains where they would basically go from like mapping horses onto zebras or zebras onto horses and so on, like basically inserting certain style from one from images of one domain onto images of another domain. So what we did, we picked up a generative adversarial network again. Actually, we used a cycle gain that consists of two gains. So the cycle gain is something that can help us moving from domain A to domain B and also moving back from domain B to domain A. So it consists of two gains essentially that are trained. And one of the benefit it has that it does not rely on paired data. So you can have, let's say, 1,000 images from domain A, 1,000 images from domain B. These don't have to be paired. So we did not have to pick up only those images that were from the same individuals. They were completely unpaired data. So here is something in terms of the results. So on the left side, we see an image for a healthy individual. There is no atherosclerosis, no accumulation of plaque. After the gain was trained, the gain was able to insert the plague. So we call it the pronounced disease case. So this is now completely, synthetic image, generated image with gain. We show this with the help of the SSIM map as well to see where the model has been able to insert the disease or the plague only in the intima media region or the wall region, not in other region of the tissues. So we see that there are quite few changes have occurred in other part of the tissue. Okay, a lot of changes are occurring in the wall region. So here is a much organized way of representing this. So we see in part A, we see healthy individuals, no intima media thickness or very small value of intima media thickness. On the right side, we see images of those individuals where the intima media thickness is quite huge, greater than 1.1 millimeter in our case. So it represents that there is a lot of plaque already accumulated. We train again to move from this domain A to domain B. So we show this in part C, and then we plot the similarity maps in terms of the SSIM. So a smaller value of the SSIM shows that there is a lot of difference. So the green is showing that the two images are the same. So we see that no differences occur in other parts of the image, which basically shows that no differences has been induced in other part of the tissues. The only major difference is that the model has learned to introduce is in the IMT region. Okay. Of course, there are some differences here on the top left. We can see this was something that we wouldn't have wanted, but the model has somehow picked and introduced some differences here as well. So yes, this is another example here. Again, in the intima media region, we see a lot of changes. In other region, we don't see much region. But I said in the top left, we do see that the model has introduced some changes that were kind of, undesired, or this was a behavior that wasn't our goal from the model. One thing that we observed with this was that the region with high contrast were quite helpful. Okay, so if you look at image C, the right side of image C is with high contrast, while the left side is with low contrast. In the transform image, we can see that the model has been able to introduce this plaque only in that area of the image where the contrast is quite high. So on the right side, we see a lot of differences in domain A, domain B. Okay, but on the left side we don't see much difference. So it does show that the high contrast of the ultrasound image itself was quite helpful for the model. So the model was able to pick those regions and kind of insert the plaque or the behavior of the plaque in those regions where the contrast was high. We also did some reverse generation, so not just moving from domain A to domain B, but also moving from domain B to domain A, basically moving from disease scenarios to healthy scenarios. This was only for the sake of completion of the model. So this was some work done in collaboration with Christer, Robin, and Johannes. Also, I would like to acknowledge the funders here, especially the Swedish Research Council, which funded this research, and the Kempe Foundation, which funded my stay over there, and Umea University, which provided the platform for doing this research. So moving to the next project here, which is on abdominal CT to abdominal MRI for liver segmentation. So again, we will see some nice application of gains here. So now we are moving from CT to MRI, and just let's interpret this CT to MRI as two different domains of imaging. Why do we want to do it? Why do we want to generate this synthetic MRI? Because we somehow want to benefit from, uh, increasing our data set size so that it can help us increase the liver segmentation performance. So what we do is we treat this as a cross modality image transformation problem, okay, moving from CT to MRI. So here we again use a cycle gain model combined with a segmentation network, and this is something that is referred as the SNET architecture, which is basically using the cycle gain along with the segmentation architecture. And again, in this case, the data is unpaired. We do not use any paired data. So the two set of training data that we have used are not basically paired. So these are for different individuals. So the overall architecture has two parts. One is the SNET, one is the unit. So the SNET is basically using the cycle gain to generate the data, and then we train a unit to perform the segmentation. So here is the overall architecture of the SNET part, which is again comprising a cycle gain. So the cycle gain has two gains, so two generators, two differentiators. Okay, it will generate some data for us, and then we use once the model has been trained, we use the generator G1 only to generate our MRI image data. Okay, here in terms of the data set, we use the chaos data set, quite popular data set. So it has CT images of 40 different individuals. Okay, so we pick those CT images, and it has MRI images as well for different individuals, I guess again 40. Once we pick those 40 cases, we extract. So these 40 cases correspond to 120 DICOM files. These are 3D files. We basically extract 2D images from these 3D DICOM files. We could only use 20 files out of these. Okay. So once we extracted the images, we ended up having almost 2800 images. So of these, we just use then pick further, refined our selection and picked only 2000 images because we needed those images where the liver is quite clearly visible. For the MRI data, again, for 40 cases, we ended up using 623 images extracted from 20 files. Okay. So yeah, in terms of results, here we are looking at the results for the model. So we are doing a transformation from CT to MRI. Okay, these are some simple images. Again, on the right side, we see some CT images as input and some MRI images as output. So the MRIs are completely synthetic here. What benefits does is bring? It improves the segmentation, but we look at it in a while. Does it bring some artifacts? Yes, it does. We looked at that possibility, whether the model also brings some kind of artifact in the liver part. We were able to notice some minor artifacts, and that can actually also affect the performance of the liver segmentation as well. Uh, I mean, we did try different training mechanism, but like we couldn't get rid of these. Okay, so does it bring any improvement despite having these minor defects? Yes, it does. What we noticed was when we generated those MRI images and now use them along with real data to train a unit segmentation, we could see some improvement in terms of the dice score, in terms of the intersection or union score. Okay, so but this is an interesting behavior here. Here we are increasing the size of the data from 350 to 704. Okay, we see some improvement again from further increase to 1000. We see some improvement, but any increase beyond that is kind of fluctuating the performance in terms of the dice score. So it means that we cannot just infinitely introduce synthetic data into our data. But overall, we can improve the performance of the segmentation by introducing some synthetic data. Okay, so here we see some performance for real images only. When the model is trained with real images only, we see the output on the left side, the first column of this figure. So compare it with the third column that contains the ground truth. And once we train the model with the real plus synthetic data, we can clearly see that the segmentation is now much more closer to the ground truth. Okay, so these are a few selected examples. So for example, here is quite obvious in the first row. Even in the second one, it's quite obvious that the training with real plus synthetic data has definitely improved the segmentation. For the third one, again, very obvious. For the fourth one, yeah, again, even if it's not exactly the same, but much better when we compare it with the training of the real images only. And likewise in the fifth case. Okay, so I hope we got time. Yeah. So next one, I'm going to talk about doing super resolution with gains. So super resolution is an idea of moving from low resolution domain to high resolution domain. So now you can see that we are actually defining the problem of super resolution as a domain to domain translation. So here we treat the data as the data that belongs to the low resolution domain. So let's say we call it domain A. And then we have some more data, which is a high resolution data. We call it domain B. Okay. So essentially we are training a GAN model to move from domain A to domain B. So again, it's treated as a domain to domain translation. So here we have done, so there were some techniques for super resolution. What we did was we introduced some changes to those architectures. For example, one of the key change that we did was that we did a multi-scale, a multi-layer or a multi-stage upscaling. So instead of just applying the upscaling in one stage, we did an iterative upscaling. Okay, and likewise, instead of extraction of features at a using a fix filter, we rather extracted the feature using filters of three different sizes. Okay, and then we evaluated this on four different modalities of data. We used retinal images, we used the skin cancer images, we used the Braid's data, which is basically for brain MRI, and then we used some ultrasound images as well. So here is the demographics. If you look at this demographics summary, none of this data is actually quite huge in terms of the number of images that we are using. So for the retinal images, we ended up using almost 400 images, not so huge. For the skin cancer data, we are using around 500 plus images. Again, not so huge. For the brain MRI, we used around 400 images. Again, not so huge. And for the ultrasound imaging data, again, we use 400 images for the training of this model. So in terms of the pipeline, as I said, so we extract the features at three different scales. Then we perform an upscaling followed by another phase of upscaling before we could perform the reconstruction to get the constructed image. So in terms of the first part of this architecture where we do the feature extraction, we can see that we are doing extraction using three different sizes of the filter, a filter of size 3, a filter of size 5, a filter of size 7. And we believe that it helps in kind of, extracting features that might be missed by other features. And at the end, we just combine these three set of features and concatenate them. The two upscaling is just using a CNN for upscaling followed by another CNN for upscaling. So essentially we are doing a 4x upscaling before we can reconstruct the data. So in terms of results, if you can see on my screen, so here is then high resolution image on the left side. There are a few techniques that we are comparing with. So we can clearly see that the uh, upscaling or the conversion to the high resolution by this particular method is resulting in an image which is relatively closer to the original high resolution data. It is somehow able to preserve quite nice details, but you might also notice, I'm not sure how much clearly visible it is on your screen, but you might also notice some blurring effect, although the structure of the veins, the structure of the artery vessels are still, quite nicely retained. This is an output on the skin cancer images. Again, we can see that compared to some other methods, our method is kind of able to retain quite nice details, much closer, much similar to the original high resolution image. This one is quite interesting for the brain MRI. We can clearly see that it has much lesser blurry effect. It has much nicely recovered the details of the region, okay, quite nicely. And this one is for ultrasound images, the more out of the four modalities, the most challenging one. We didn't see much improvement actually here. Again, this was worked with my wonderful colleagues, Rafiq, Wakar, and Dr. Shweb. Okay, so now I'll just quickly talk about the neural diffusion models for prostate cancer images. This is something that we started recently at Stirling here. So, prostate cancer, as you might know, is a cancer that is, related to men. The prostate cancer UK website says that out of 8 men, one man is going to get this prostate cancer at some stage. Surprisingly, if you are an individual, a black individual, your risk of getting the prostate cancer is double. It's quite a challenging one to detect in terms of clinical detection because there are no obvious symptoms for quite some time. Even if you have developed a prostate cancer, the symptoms that it has, the symptoms that it brings can be easily confused with, urinary tract infection or difficulty in, releasing your urine. So it might not be very obvious. So one of the most prevailing, I guess the second most prevalent cancer in men, and as you grow, so it increases, the tendency of this cancer increases with age. Commonly, it is studied with MRI imaging modality. So if someone has got a prostate cancer, what is the next stage to do in terms of the treatment? So the doctors would look for possibilities of radiotherapy or chemotherapy. But the radiotherapy is a really sensitive treatment technique. You only want to treat the cancerous tissues. You do not want to apply the radiotherapy to healthy tissues of the body because it can just damage the healthy tissues. So a key here is the segmentation, and not just segmentation but rather precise segmentation of the prostate. So again, can we use deep learning? Yes, from our various success cases of deep learning segmentation techniques. Yes, we can use it, but we need data. And data is something that is image data is something that is really scarce in this case. One of the common challenge is if we don't have enough data, if we don't have a good model, the model trained on smaller data kind of end up in over segmentation, which is quite a problem. So here we move to the diffusion models to generate more data that can help us, train a segmentation model in a much better way. So we use a mass condition denoising diffusion probabilistic model. So here is the data that we have used is from the university. It's publicly available. Anyone can apply for it, fill the form and get access to the data. It's quite a huge data. It's around 200 GB, maybe the first large scale data related to prostate cancer. And it was released just last year. So it presents prostate cancer studies of 400 plus individuals. Okay, so what we did, we picked this data, it trained a diffusion model to generate more images. We used those images along with the real images to see whether it brings some kind of improvement in the performance of segmentation or not. We also then evaluate the model on additional data that comes from the medical segmentation decathlon. It is a data for 32 individuals. It presents 32 cases. So our hypothesis here is that when we started this work, yes, with this motivation that we do expect, based on our previous studies, we do expect that if we use a diffusion model to generate more data, we might end up, bringing some improvement in the performance of the segmentation model as well. So here we use the NN unit segmentation model because it's kind of, quite popular these days. Uh, so our hypothesis that we ended up was that a moderate introduction of synthetic data can actually improve the segmentation performance. So I won't go into many details because this is work in progress and under review as well at one of the conferences. But here are some outputs. So I'm not sure if this is so easily understandable, but we can clearly see, if we interpret this, we can clearly see that the when we train the model with augmented data, we see some improvement in the segmentation, but it becomes much more clear in terms of quantitative metrics. So we evaluate this with the help of HD 95 metric, which is a distance metric. So in short terms, like the lower value is better. So it basically is a distance measure. So we can see that for baseline NN unit without any augmented data, here are the values for HD95 for these 5 cases. And for the models when trained with the augmented data, we can clearly see improvement in the HD95 score. We then evaluate this model. So we have trained the model with the data that we generated from the diffusion model. Okay, and the diffusion model used data only from the Lund probe dataset. It did not see any data from the MSD. And now we pick up this model and evaluate it or cross-validate it on the MSD data as well. So we can see that it has reduced the false positive by almost 23%. Okay, so interestingly, it didn't see any of this MSD data before, so there is some indication that yes, so it is learning something, that can be, expanded to other data sets as well. So here I end up my slides, and there are some useful references. Here is my LinkedIn as well, my email, my web page. So yeah, now let's move to question answers.